Ayubovan Duepute, and this is about grade 10 science, and we have been talking about chemical basis of life, and this is as per the Sri Lankan government school syllabus, and we will talk about nucleic acid element tests today. We already know what is nucleic acid and what is the difference between RNA and DNA and its different functionalities. Am I right? DNA is doing some genetical works while protein synthesis is happening in the RNA. We talk about in our previous video session, and this is going to be video number 13 of chapter number 1. And what we will learn, we will talk about nucleic acid elements test. What are the tests we are going to do? Carbon test, hydrogen test, oxygen test, and nitrogen test. And we want to understand in this nucleic acid whether the carbon is available, hydrogen is available, oxygen is available, and nitrogen is available. Let's get into the subject and see those things. Nucleic acid contain mainly carbon, hydrogen, oxygen, and nitrogen. These are the main things that nucleic acid got. So we will conduct below mentioned activities to confirm the presence of those elements. Right. The first test that what we are going to do is identification of water as a constituent in food. Now when we are talking about that, hydrogen and the oxygen is available in the water. Those are the composition, right? Hydrogen and the oxygen. Now if your food has hydrogen and oxygen, that means water, that says hydrogen and oxygen available. Now identification of water as a constituent in food. For that, what we want to do, we want to have meat, one egg shell, and some plant leaves, and some crucibles we need. Not only one, some crucibles, we might need 3 crucibles to do all the activities. The second step what we are going to do is grind or crush meat. Grind or crush the meat, eggshells, and leaves in their dried form separately. In the dried form, you're going to crush or grind this meat, the eggshells, and the leaf. I did crushed in front of you. It is just like that. And then after that, what we are going to do, let's see, put them separately into crucibles and heat them just like that. Okay, now in 3 different crucibles, meat, eggshells, and the leaves are getting heated. And after some times, what we are going to do, during heating, what happens is hold a glass sheet above the crucible. In all the 3, meat, leaves, and the other one is about the eggshells, right? During heat, hold a glass sheet above the crucible. After some times, the water vapor, a kind of a vapor will appear like that. You're going to turn that one, and then you're going to take that one to one place. And blue colored anhydrous cobalt chloride you're going to put. See what is happening? Once you put the blue colored anhydrous cobalt chloride, it will change into a pink color. It will change into pink color. So that says during the heating and hold a glass sheet above the crucible. The second experiment what we are going to do is we are going to put another solution on top of it and check for it. Now this time we are going to put white colored anhydrous copper sulphate. When you are putting white colored anhydrous copper sulphate, it turned into blue color. Those 2 experiment is giving us a clue telling that it is confirmed that water is formed on the glass sheet. That particular water is becoming the color pink with one solution, and another solution it is becoming blue. So it is confirmed that water is formed on the glass sheet. Therefore, the food that is used for the experiments contains water as a constituent. Therefore, the food has hydrogen and oxygen. Now let's do another test for carbon. Identification of presence of carbon in biomolecules. For this one, we need several cubicles like this, spinach stems and piece of fish. And then after that, we need some chickpea. As earlier, we are going to do the same thing here as well. Make a pulps by crushing all above materials separately. We are going to crush it also just like this. Now once we do that one, what we are going to do, put them separately into crucible and heating well. Here also we are going to do another kind of an experiment. This time we are not going to take the glass. This time we are going to take something else, that is a piece of paper. The final residue obtained should be rubbed. Now this much of small portion is available. We are taking that one and we are rubbing like this on the sheet of paper. See what happened? Yes, lines drawn due to coal is observed. Due to the coal presence, the line is appearing just like this on the sheet of paper. Right, that says it can confirm the food that is used for the experiment contains carbon. So that is an interesting experiment also. Now we came to know hydrogen, oxygen and carbon available in the food what we consume, and the next one is about nitrogen availability. So identification of presence of nitrogen in biomolecules. To do this one, we need 2 test tubes, solution of sodium hydroxide, solution of copper sulphate, egg white and piece of fish. 5 things we require. Now what we are going to do, we are going to crush the fish thoroughly, add water and mix it well. We are crushing the fish like that, and we're taking a container and we are putting that one inside that container and we are mixing that one with the water just like this. It got mixed up well in front of you. After that, what are we going to do? We are taking that solution and we are filtering the solution. We are taking another container and taking the filter like that, and we filtered it. Now the filtered fish solution also there with us. Now what we are going to do, put 2 milliliter of fish extraction and egg white into separate test tube like this. Now we are going to put egg and the fish extraction into 2 milliliter. That is the amount that we are going to take, and it is in front of you. Now add equal volume of sodium hydroxide. We are putting sodium hydroxide. Now add few drops of copper sulphate to it. See what is happening? It turns into purple color. Adding equal volume of sodium hydroxide and adding few drops of copper sulphate to it. After that, it turned into purple color. So the purple color appears in the solution. That why it is appearing. So purple color appears in the solution, and this confirms the presence of protein in food. As nitrogen is the constituent of proteins, it is confirmed that the above tissue contained nitrogen. So now we know that nucleic acid elements test we did for the carbon and for the hydrogen, oxygen, and finally to the nitrogen also. Let's meet you in another session to talk about water. Until then, bye bye, take care of yourself.